Hello, I'm Dr. Tim Sandle and I'm presenting today on the Lister Institute and Bioproducts Laboratory, both of which represent laboratories that have been based in the Elstree Aldenham area for well over 120 years. And I've subtitled this The Story of Plague, Smallpox and Life-Saving Medicines. So before we go too far, it's important to pause and have a look at the life of Joseph Lister, or the Baron Lister of Lyme Regis. So Lister was a pioneering surgeon based up initially in Edinburgh, and he's perhaps best known for advances in antiseptic surgery, particularly with the use of carbolic acid. So before Lister, surgeons would regularly share surgical instruments between themselves and between patients, and there was no attempt to decontaminate those. Lister was one of the first to realise that this was actually a means of disease transmission, and he recommended the treatment of the surgical instruments in carbolic acid. Lister is also known as the chair of the first medical research charity in the United Kingdom, and this became the basis of the Lister Institute. So the Lister Institute of Preventative Medicine was established in 1891 as the British Institute of Preventative Medicine, and this was based in Chelsea. A little while later it merged with the College of State Medicine to form the Jenner Institute of Preventative Medicine, named after Edward Jenner, who did the initial work on smallpox and the use of cowpox as a substitute vaccine. The name was later changed to the Lister Institute of Preventative Medicine in 1902 and was established as a charitable and not-for-profit organisation. So although while the Institute was based out of Chelsea, the particular research direction it was going into required alternative premises. And the reason alternative premises were needed was that a ready supply of animals were needed for the early vaccine work. So the Lister Institute selected Dagger Farm. So Dagger Farm, based in Aldenham area, was a farm of 35 acres, and Lister took this over, and this was to develop the only institute of its kind in the UK, and it quickly established a reputation that was equivalent to the Pasteur Institute in Paris or the Rockefeller Institute in New York. And as I mentioned before, it was reliant upon charitable donations but also some government grants, including one from the Colonial Office, which I'll explain a little bit about in a moment. So just to put this into perspective, you can see the early siting of the Lister Institute around the turn of the century. So the Institute here was established to prepare and supply special protective and curative materials, mostly vaccines and antitoxins, designed to treat people with infectious diseases. And for this they needed animals, because animals could be injected with pathogens. The animals would hopefully develop antibodies, and then the antisera could be collected. Another area that was fairly pioneering for the Lister Institute at the time was with plague research and the ways that plague is transmitted, such as bubonic plague and pneumonic plague. And plague was particularly devastating in Asia, and in 1905 it alone killed 500,000 people in India. So this is one of the reasons why the Colonial Office gave particular support to the Lister Institute at the time. And one of the activities that took place was paying people in India to collect rats that had died from plague and send those back to Elstree for research, and that was led by Dame Harriet Crick, who became famous for her research later on rickets. And it also saw one of the last plague fatalities in the United Kingdom, when in the 1930s An unfortunate laboratory technician was working in Queensbury Lodge, which was then the site of the laboratories, and they contracted the pathogenic organism, went back to the Lister cottages and passed away. The other thing that the Lister Institute was particularly well known for was its role in development of the smallpox vaccine and its contribution to the worldwide eradication of smallpox. And in particular, the Lister Institute developed a freeze-dried smallpox vaccine, which meant that it was fairly stable and could be sent around the world. And for this they used a particular strain that was derived from the battlefields at Crimea, and this, after processing, became the Lister strain. And this was the most widely used vaccine in the world from a period in the late 1960s through to the mid-1970s. And so important was this vaccine that it was also sent as on humanitarian grounds to the Soviet Union, because it was seen in everybody's interest to eradicate smallpox worldwide. Okay, so before moving on to BPL, I think it's important at this point to pause and look at blood plasma. So blood is made up of 55% plasma, which is this yellowy straw coloured fluid, 45% red blood cells, which pass oxygen around the body and take waste and carbon dioxide out of the body, white blood cells which fight infection, and platelets to help with the clotting mechanism. And an element of plasma contains over 500 different proteins, and some of these proteins are of a therapeutic value, and perhaps three of the most important groups are albumin, which is a globular protein made by the liver, and when that's used for a patient it can help to restore lost blood volume. Then there are gamma globulins or immunoglobulins that contain antibodies to help people fight against bacterial and viral infections, and then there are the blood clotting proteins that can help to address conditions like haemophilia. So plasma proteins can be used to fight a number of life-threatening diseases, genetic conditions, and to help people live a normal life. Now, blood products, which are products made from the plasma fractions or the various protein elements, were advanced by a man called Edwin Cohn, who developed a fractionation process, and he discovered that if you mix plasma with ethanol at cold temperatures, then over time different proteins of different molecular weight are eluted, and the ones of interest can be collected and turned into therapeutic products. While this initial work was going on, there was also realization that plasma, most prominently the albumin form of plasma, could be used to help people who have been injured in battles or through associated damage in World War II. So this could help to treat shock or to restore loss of blood volume. And this led to something called the Plasma for Britain campaign. And here people in the USA were donating their plasma. It was freeze-dried and sent to Europe, particularly the British forces. And this interest, or this process that was going on, became a research area within the Lister Institute as well. So this is the start of the UK research. And the Blood Products Research Unit was formed, and this was part of the Lister Biophysics Department. And this was backed by the Medical Research Council. So in the early 1950s, this kind of interest in the possibilities of blood products began to emerge. Now, this research unit transformed into the Blood Products Laboratory, and this was established as a standalone centre within the site occupied by the Lister Institute. So BPL was formed with a government grant of £175,000, and it was initially run by the Medical Research Council with a remit under the Ministry of Health to supply human blood products to the NHS. And one of the early developments was the world's first Factor VIII, which was pioneered by the leading researcher of the time, Ralph Ambrose Keckwick, and Factor VIII was designed to treat a male hereditary disease, haemophilia, where the person's blood is no longer able to clot effectively. The early years of BPL and this Factor VIII development was enhanced by a grant from the US of £3,400 from the Rockefeller Foundation. And the early scientists at the time decided to take this money, and they invested in an oil turbine, an equilibrium ultracentrifuge, and this was designed and made in Sweden. And it proved so effective for the purification steps for creating blood products that it was actually actively used for 30 years. And this very centrifuge is now on display in the Science Museum in London. So after Factor VIII, the first major non-coagulation factor to be developed was a immunoglobulin, and this was used for treating rhesus negative mothers in the 1960s to prevent the mother's immune system from acting against the baby she was carrying. BPL also pioneered radioimmunoassays to help screen for hepatitis, and further immunoglobulin work continued. So there are various immunoglobulins developed to help the body fight different diseases such as tetanus and rabies, and also a very successful normal immunoglobulin was formulated to help people who were suffering with immunosuppression, particularly primary immunodeficiency. BPL continued to expand, and into the 1980s. By that time it's been run by the North Thames Regional Health Authority, and then by the Central Blood Laboratories Authority. And it included some more disparate elements. So there was a specialist coagulation factor research base in Oxford called the Plasma Fractionation Laboratory, and this ran from 1967 to 1992. There was a laboratory specialising in blood grouping reagents, the International Blood Group Reference Laboratory, which is based in Bristol Hospital, and there was also various diagnostics and serological tests being worked upon. Around that time also BPL got much bigger, and it purchased a nearby orchard and expanded, and it also developed a process for ensuring that the products were free from harmful viruses by introducing heat inactivation, and it was the first company in the world producing blood products that developed this process. Now, plans to discuss and to expand BPL began in the 1970s, and this was based around the objective of the UK becoming self-sufficient in blood product, and this was something that was initiated by the Labour government at the time. Now it took a while to secure the funds and to get the various agreements to build a new factory, but that took place, and the kind of wedge-shaped building that could be spotted from different directions was built in the 1980s. And this process began by the then Minister of Health, Norman Fowler, laying the foundation stone, and the eventual building was opened by the Duchess of Gloucester on the 29th of April 1987. Into the 1990s, and Blood Products Laboratory was renamed as Bpo Products Laboratory, and this was mostly due to the advent of the internal market in the NHS, where hitherto BPL products had been passed freely to hospitals, they now developed an internal charging mechanism which was designed to create more efficiency. BPL was then also absorbed into the National Blood Service, which exists today as NHS Blood and Transplant. A major shockwave took place in 1996 when it was discovered that new variant CJD, which is a prion disease, could be passed from infected cattle to humans through the consumption of beef, but there was also a concern that it could also be passed through blood plasma. So this is because of the infectious agent, a folded piece of protein that couldn't be guaranteed to be eliminated wholly from plasma. So this led the government to instruct BPL to move to non-UK sources of plasma. So it was decided to purchase plasma from the USA, which obviously created a number of logistical problems. Now, plasma safety is a very important element, and every plasma donation from the US needs to be tested for markers for potential bloodborne pathogens. So this includes serological tests for viral markers of concern, particularly hepatitis B, the HIV virus, that's responsible for AIDS, hepatitis C, human T-cell lymphotropic virus, also paravirus, and there's also screening for one bacterium, which is syphilis, which is undertaken by assessing for syphilis antibodies. And if any donation tests positive for any one of these pathogen markers, then it is excluded and it never forms part of the manufacturing process. BPL has also undertaken some other innovations. So for improved patient safety, it again pioneered the solvent detergent process in immunoglobulin manufacturing, which was again targeted to eliminate viruses. There were developments with coagulation factors to make these more applicable to a wider range of bloodborne diseases, such as von Willebrand disease, where people need additional factor VIII to deal with rarer types of blood clotting diseases, and also for haemophilia A and haemophilia B. There's also a product for a very rare form of coagulation factor disorder, which is to address factor X deficiency. There was an alternative immunoglobulin that could be delivered under the skin or subcutaneously, and there was an improved intravenous immunoglobulin that is generally recognised by a number of the majority of medics as one of the superior blood products on the market. So BPL Today has had a very kind of mixed history. So it arose from the Lister Institute. It was then run by the Medical Research Council. Then it was run by regional health authorities. It then fell under its own self-governing health authority. Then it became part of the National Blood Service. Then it was run directly by the Department of Health. Then it was co-owned by a venture capital firm called Bain Capital and the Department of Health, which is through the Cameron government's attempt to sell off a number of state assets. Then it was sold to China and owned by a company, Creat, and as we speak now, it's undergoing a merger with an Italian company called Kedrion. And it's important to note that BPL will be celebrating its 70th anniversary in 2024. And then BPL Today, you can see in the purple the number of range of different products that are manufactured at BPL. So it's principally these coagulation factors to address bleeding disorders, immunoglobulins to treat either specific diseases to help the body to fight things like tetanus, or it's there to address conditions like primary immunodeficiency. And then there is albumin, which is all about rebalancing the body. Today at the Elstree site, BPL employs around 1,000 people, and these are divided between manufacturing, research and development, quality and commercial as the principal departments. All products are still manufactured only from human plasma, and there's a very complex process which begins with fractionation, this whole protein separation thing, and then into the purification. And all of the products are sterile medicines and are produced by aseptic processing. Okay, so this brings the presentation to an end. I hope it was of interest, and I'll be happy to answer any questions that anyone may have. So I've been Dr. Tim Sandle. Thank you very much for listening.